This white paper podcast is brought to you by DIA and LARVOL. LARVOL supports immuno-oncology research and treatment by providing custom intelligence around predictive biomarker and diagnostic development, as well as animating a complete clinical trial database available today. According to the World Health Organization, cancer was the second leading cause of death, accounting for 1 out of every 6, or 9.6 million deaths in 2018. Cancer care is complex, and most patients are treated with a combination of surgery, radiation, and drugs. New drugs, addressed as immunotherapy, are designed to help the patient's own immune system to fight cancer. Extensive research is ongoing and introducing these drugs into clinical practice. One class of drugs is called immune checkpoint inhibitor therapy, which helps the body recognize and target cancer cells. The recent approval of multiple immune checkpoint inhibitors has brought us to a critical juncture in clinical research, changing the standard of care in many different cancer types. I am Chris M. Slowecki, Senior Digital Copy Editor for DIA. Today, it's my honor to welcome and introduce two colleagues in the dynamic and critical field of cancer immunotherapy. Kinisha Gala, who serves as project owner of oncology for LARVOL, and Professor Charlotte Moser, radiation oncologist and expert in cancer combination therapies and drug development, who serves as Vice President Clinical Development at Galera Therapeutics, Inc. Thank you both for joining us today. Charlotte, let's address that first question to you. What major changes or trends have emerged in cancer clinical trial design over the past 10 years? And then sort of the other sign of that, what are some of the more challenging aspects of clinical trial design that still exist in oncology research?
The main thing we learned over the last 10, even 20 years is that cancer types like breast or lung or colon cancer are all very different diseases and consist of various subtypes. So breast cancer is not one diagnosis, and it's very important to know what is the biological or genetic subtype of that breast cancer, because they now are various and different treatments for specific subgroups, and they relate to a very different prognosis. But overall, most of them, they need various treatments, chemotherapy, but also surgery and radiation, for example, in an order and in a timing, which makes cancer care quite complex to oversee in clinical practice, but especially when you want to design a clinical trial and observe benefit for a new drug, for example, you have to take into account all the other treatments who are given as a standard of care for that type of cancer. So I see overall that we are working in far more specific subgroups of the disease, which means automatically smaller cohorts. And the challenge is to monitor not only the treatment you add, but all the other treatments associated given as well. And then look not only at the outcome, if the treatment is working, your cancer is cured, but also what kind of side effects you provoke during that treatment, but as well after treatment. Because in clinical practice today, when we need to decide what treatment we're going to prescribe or what series of treatment we're going to prescribe, we have to take into account not only what we can gain in survival, but as well as what burden and side effects we're going to provoke. Especially in long survivors, we have to take all that quality of life compromises into account in our current choice of treatment.
Thank you, Charlotte. That begs a follow-up question, I think. How have the changes in the decisions made in clinical care driven changes in the design of related clinical trials?
So overall, I think, and that's specifically the last 10 years, that cancer care needs a multidisciplinary approach, like a tumor board or tumor team who is specialized in specific cancer subtypes, and is a team of surgeons, medical and radiation oncologists, but also pathologists, as well as imaging experts, for example, who need to reunite and look at the diagnosis, classify and stage each patient, and then decide together what kind of treatments are needed And as I said, in which order, because we cannot give all treatments at the same time. So we need to space them out in terms of not risking too much side effects because of synergies, right? So every cancer is tackled or treated or approached by a team. And within those teams, the cancer specialists have become even further subspecialized, saying we have breast surgeons or pathologists, even oncologists who only treat melanoma or specific lung cancers, which was not the case 10 years ago. But that's simply because we really need to stay on top of all the new literature and knowledge coming out of clinical trials, which are quite impressive number every month, every year, difficult to tackle in all sides of cancer care in general. So we really work in sub-discipline groups, having all disciplines on board to treat that cohort of patients correctly.
Thank you for that answer, Charlie. Kinisha, if we can turn to you for a moment. With the recent approvals of multiple immune checkpoint inhibitors, we're at a critical point in cancer immunotherapy. So, what challenges to clinical trial design are specific to immunotherapy?
Thanks, Chris. Yeah, immunotherapy is a very rapidly moving field. And as you just alluded to, there are challenges to clinical trial design that are specific to immunotherapy. The first thing that comes to mind is that the timeline for immunotherapy is quite a bit longer than what we're used to with chemo or targeted therapies. And by this, I mean evaluating response. We often can't do it the same way that we're used to. The short-term efficacy endpoints, like progression-free survival, for example, may not be adequate surrogates for the long-term efficacy endpoints. Similarly, on the toxicity side of things, we're finding that immune-related adverse events take much longer to manifest, maybe up to a year, or, in some cases, maybe even longer to manifest. And this longer timeline is difficult for clinical trial design. Longer trials are more time-consuming, expensive, and honestly not always feasible. And so I think one thing that we really need is better tools to predict response, maybe even better tools to predict which patients deserve longer follow-up, also better tools to predict what toxicity certain patients may experience. Kind of following up on that, one thing that immediately comes to mind when you're talking about predicting responses is biomarkers. And we still don't have a complete picture of the use of biomarkers in immunotherapy. For certain cancers, maybe like lung cancer, we do know that PD-L1 expression does predict response to immunotherapy, but there's still so many unanswered questions, even in lung cancer. For example, if we have biomarkers taken from a biopsy sample years ago, is that still relevant as eligibility criteria for entry into a particular clinical trial? And the lessons we've learned from lung cancer, we're finding that they're not necessarily easily translatable to other cancer types. This kind of goes back to what Charlotte was talking about in the previous question. So to kind of sum it up, I think that the longer timeline makes clinical trial design in immunotherapy especially difficult. And we still have a lot of work to do in terms of predicting response and in terms of how to best use biomarkers. And one last point I want to make that's specific, well, maybe not completely specific to immunotherapy, but what we're finding with immunotherapy is it's often given and designed in combination with other drugs, in combination with chemo, for example. And so this makes understanding toxicity a significant challenge. We now need to evaluate how the toxicities of multiple drugs are working together in the patient, whether it's synergistic, there might be, a protective element. And so that's an additional layer of another challenge in terms of designing clinical trials for immunotherapy.
Can I add on that? Because I think it's really important what Kinisha is saying now in terms of adding on in combination. But it's really that they can combine, give a better result, but they also combine their toxicities, right? Can be in 1 + 1 is 2, but can also be a synergy going up to 3 times more toxicity when you combine them, right? So there is a risk when you give 2 drugs or 3 drugs, and it's not always adding up like 1 + 1. It can add up 1 + 1 becomes 3, right? And that follow-up is always the critical point in clinical trial design because it costs a lot of money. And especially when you want to test a new drug in an early phase, you do not have that kind of money to monitor patients so closely for safety as well as such a long time after. And those means are simply limiting that knowledge we all need so much to conclude about new ways to go and new standards in new cancer care.
Charlotte, that was both right on time and extremely on point. Kinisha, I want to stay with you for just one more moment, but I want to face forward a little bit. We have a next generation of immune modulators. We may not even know fully their capabilities or what they may look like. How does this emerging next wave make figuring out clinical trial designs for oncology trials even more complicated for folks like you?
Yeah, thank you. That's a great question. I mean, now we're, going to be dealing with a patient population that's already progressed on first generation IO. The immediate concern that comes to mind is, again, toxicity. So toxicity remains a significant concern. We're now considering toxicities associated with IO combinations. As Charlotte just mentioned, it's always possible that the side effects are synergistic, 1 + 1 = 3. And we're effectively blocking multiple pathways of our own body's immune system. So I think toxicity is a big concern when we're talking about treating patients who have already progressed on IO with even more IO. But specifically in terms of clinical trial design, what we'll see as we get better at using biomarkers to predict response is we might get into a situation where now we're designing clinical trials where we're making use of multiple biomarkers. And this leads to potentially even smaller patient populations that are eligible for these clinical trials. And so for this complexity in particular, there have been relatively new clinical trial formats like platform trials. So these would be like basket trials where there are multiple histologies and a single biomarker, or umbrella trials where there's a single histology and multiple biomarkers as a way to accrue enough patients that need to potentially be expressing, one or more biomarker. And one point I want to make particularly about these platform trials is we need to start paying attention to the non-responders as much as we are paying attention to the responders. So typically we use these trials as kind of a treasure hunt to find the patients that are responding to the drugs. But I think we should also aggregate and organize the data of the non-responders as well. As I said earlier, I think one thing that can really help immunotherapy and help clinical trial design is the ability to predict which patients will respond. And I think in order to best do that and in order to best model response, we need to understand why patients are responding and also why patients are not responding.
I would like to add here is that we too often think in treatments like surgery, right? We give immunotherapy and it works or it doesn't work. However, immunosystems and immunotherapy can be far more flexible in the sense that in the beginning of a disease, it cannot work, but because of changes within the tumor, because of other treatments given, that can change into going to work, or vice versa, in the sense that we know resistance works is that in the beginning it can be sensitive, but then the tumor is so intelligent that it starts to block and then the treatment is not working anymore. I think we have to stop looking at treatments by saying it works, it doesn't work, and look at where in the time of the patient pathway it works or could work. And that means that the biomarkers should be also seen more dynamically in saying when are they measured and what are they saying, and are they remeasured after, for example, an intervention, and is the result the same?
Charlotte, the number of available treatments seems to be growing, and yet at the same time, research has seemed to pretty conclusively prove that breast cancer, for example, isn't one disease, but is in fact a collection of smaller patient subgroups with different types of cancers. So on the one hand, the patient subgroups are getting smaller. On the other hand, the treatment options, the number of them seems to be getting larger. How is that impacting care for cancer patients? And then secondly, how is that impacting the design of clinical trials for further research for these therapies?
That's exactly what I wanted to say. In the first challenge, what is changing so fast over the last 10 years, coming with the more knowledge we have of biological subtypes and genetic backgrounds of those different cancer patients and tumor types. In general, in practice, that means that we need to specifically subtype each patient in a correct way. And then means that then we can decide on the series of treatments based on those characteristics. And that needs a specific qualified team, right? And the team, I worked in the breast unit for a lot of years, and it's really, really a different atmosphere working with a specific team, all doing the same, around the same type of patients, the same type of literature, the same type of research. Doing that work together is completely different than in a general oncology department trying to treat as well melanoma as breast as lung cancer, right? You do not have the same overview and expertise you really acquire in such subunits when you're so specialized and focused on that population. But that Kinisha said, yes, there are, while doing research and trials, there are far less subjects to encounter, to include in trials if there is specifically, for example, triple negative patients who do not respond anymore on first-line chemotherapy. That's a very specific subgroup. And that means that every trial you have to lift throughout different institutes to have enough subjects together to be conclusive. And that comes to an operational hurdle as well as a data sharing hurdle, especially when you go overseas to try to reunite European centers with US centers, for example. And I hope is going to be there is that we will have far more data tools and computing tools to be creative with data generated across institutes and across countries. So we can, although the number of subjects are limited seen per site, that we still worldwide can accrue or collect enough data around that topic to be conclusive.
Thank you very much, Charlotte. Kinisha, if I could address the closing question to you. Looking back in the light of all these challenges, in the context of all these emerging drug targets and therapies, could we ask you to share some best practices for clinical trial designs in this field?
Clinical trials are, inherently limited. You're looking at a very specific patient population, and there's always a question of, how does the data gathered in this limited population, how does it translate to the general population? Particularly for immunotherapy, as, Charlotte and I alluded to earlier in the conversation, it's worth it to have systems in place for long-term follow-up of patients starting right at the phase 1 trials. As Charlotte said, that's extremely expensive and it's not always feasible. So I think one thing that we really want to work towards, and I mentioned this before, is being able to predict, maybe even predict the subset of patients that are high risk, where it would be worth doing that long-term follow-up. And of course, getting better at our ability to predict response and, potentially even toxicity. And I think one thing that's really key to make these predictions is aggregating clinical trial data from past clinical trials. Historically speaking, there have been multiple efforts to aggregate clinical trial data, but I think these have largely focused on tools for clinical decision-making, for clinicians, or even patient-focused tools for helping patients find the right clinical trials that they might be eligible for. And I think there is a lack of collecting and aggregating clinical trial data for the specific purpose of informing new clinical trial design. This can be used for things like creating historical control arms, ensuring adequate sample sizes, which is really important, and also, just simply reviewing the appropriate references. So you have a really good understanding of the field, the standard of care as you're going into designing your clinical trial. And I think one step beyond that, once we're, able to aggregate this clinical trial data, we can then use higher level computational techniques, machine learning, and higher level data analytics to make these predictions about response and to make these predictions about the higher risk patients that might deserve longer term follow-up. And I think that can really make a big difference in immunotherapy, but also in oncology clinical trial design in general. This is something that my company, LARVOL, is really enthusiastic about working to consolidate clinical trial data in a curated fashion, and particularly making it easy to access specifically for the purpose of clinical trial design. And I think more and more efforts to do this, to bring the clinical trial data together and make it available for people that are designing the clinical trials, I think that's going to be key in moving forward. We're moving at such a fast pace that we need to sometimes stop and make sure we are learning all the lessons that can be learned from the trials already conducted.
Yeah, I can add there a very simple example. In a rare cancer type, you want to set up a trial. And as I said, to find enough subjects, you will need at least 15 or 20 centers to work around that. Then when you're going to make your budget, you need to calculate how much subject you would need to give a sensitive answer to your question, as well as how long they should be followed, and giving an estimation of their PFS, or so their progression-free survival, or how long they will be still alive. And all that is based on literature and looking through what kind of population you're aiming at. And it would be so wonderful if we could use all the posters, publications, and communications out there to use that and say, okay, I have this very specific triple negative patient population after second line, and I want to investigate that group, and I want to know what is in general their survival chances. So then with that knowledge in hand, I can then with the statistician estimate how much subject I will need and how long I will have to follow them to come to such an endpoint. And that means that the better I can set my budget right, the more feasible it will be to do the trial or even decide to start it, yes or no. It's just an example, but I think we should be far more creative using our literature and congresses in that sense, and not only in an educational sense.
I completely agree. And I really appreciate bringing up of an actual example of a rare disease type where it would be really useful to have a centralized database where you can access all of this information, again, for the specific purpose of designing clinical trials. Thanks.
Yeah, and it comes not only at the subtype of disease, right? So not only lung cancer, but PD-1 positive or not, as well as the treatments they already got, or in what kind of combination you're going to give it. So it's that complexity, when you want to put a figure on it, it's not easy to do by hand.
Right.
Thank you both very much. This has just been a remarkable conversation. Charlotte, thank you very much for sharing your insights today.
My pleasure.
Kinisha, thank you as well. Thank you for your part in this conversation today.
Thanks so much, Chris. Thanks for having me.
For DIA and LARVOL, I am Chris M. Slowecki. To learn more about this topic, visit our white paper library at diaglobal.org or visit larvol.com.